Well, hello and welcome everyone to today's webinar. This is Rick Import for Fisher Healthcare, and our session today is Demystifying the Development and Implementation of Molecular Tests in a Clinical Laboratory, and our presenter is Maura Aspinall, Professor of Practice of Biomedical Diagnostics at Arizona State University, and our session is sponsored by our good friends at Fisher Healthcare. Before we go any further, I just want to take just a few seconds here to be sure that everyone can hear us. If you're new to the WebEx environment, you should know that if you're hearing my voice on your computer speakers, you're all set, but you can dial into the teleconference at any time if you prefer to get your audio that way by clicking on the Participants button at the bottom of your screen, which will open the panel, the Participants panel, and at the bottom of that, you'll see a phone icon. Click on that, and that will lead you to the Connect Audio button and then the call-in box with all of the dial-in information. If you do have any comments or issues or questions, you can use the chat box for that. You can click on the chat bubble at the bottom of your screen, and that will open the chat box. And if you have any questions for Maura, you can use either the chat or the Q&A panel. The Q&A can be reached by clicking on the button with the three dots. If you'd like to send us a chat, just type into the small box right there and hit enter. We'll be providing you with links a little bit later in the session that will appear in the large box. And as I said, if you have questions for Maura, you can send them either by chat or using Q&A. Again, send your question in as they come up and type them into the small box and hit enter, and we'll take them when we get to Q&A. If you'd like to receive PACE or Florida Laboratory or a certificate of attendance for today's session, here's how that'll work. When you leave the session and close the WebEx window, you should see the evaluation open in your browser. Fill it out, send it right in. We'll be sending your certificate of attendance as a link in an email within a couple of days after we receive your certificate, your evaluation rather, provided we receive it within 24 hours. So do send it right in. If you're the person who's logged in for your group, you'll be receiving, whoever logs in today will be receiving an email from messenger@webex.com. So forward that email to those folks who joined with you so that they can access their evaluation. The link will be in that email, and that also applies to folks who have joined us on a mobile device. Watch for the email from messenger@webex.com. The recording and slides will be posted here. We'll provide this link to you in the chat box a little bit later, so you don't need to write that down, but it will be posted within a few days after today's session. And now let's move to our distinguished presenter. Maura Aspinall is a healthcare industry leader and pioneer committed to active civic involvement. She is the managing director and co-founder of Bluestone Venture Partners. Aspinall, or Maura, also heads the Health Catalysts Group, a consulting firm for HIT and Diagnostics, publishing the popular Health Catalysts Diagnostics Year in Review. She is a member of the board of directors of Abchem PLC, Allscripts, Castle Biosciences, OraSure, and Blue Cross Blue Shield Arizona. Maura is an advisor to the Rockefeller Foundation on COVID diagnostics. She is co-author on the Foundation's national reports on COVID testing, education, and policy recommendations. Maura is passionate about education on diagnostics, genomics, and personalized medicine. To that end, Maura co-founded the School of Biomedical Diagnostics at Arizona State University, the first school dedicated to diagnostics as an independent discipline. Maura has extensive experience in running laboratories. Previously, she served as president and CEO of Roche Tissue Diagnostics, president of Genzyme Genetics and Genzyme Pharmaceuticals. She's on the Health and Human Services Secretary's Advisory Council on Genetics. Health and Society was named Arizona Biosciences Leader of the Year by the Arizona Bio Association, and one of 100 most inspiring people in life sciences by Pharma Voice, and one of Women Inc.'s most influential corporate board directors. Maura holds an MBA from Harvard Business School and a BA from Tufts University. It is a delight to bring to our podium Maura Aspinall, who is actually a spectacular presenter and on top of all of those other things. So welcome to you, Maura. Looking forward to your presentation.
Well, thank you so much, Rick, for that generous introduction. What I will say is I've had the privilege of working with great people, and that has made all the difference. So it's wonderful to be here with you today. What I'm going to talk about, our official title is Demystifying the Development and Implementation of Molecular Tests in a Clinical Lab. But my working title is a little bit more fun. The simple, sensible, salient, yet still spellbinding 7 questions about laboratory-developed tests. We're going to talk about laboratory-developed tests, as most of you probably know, what we call IVDs, but we're also going to talk about IVDs and what those mean. But before we do that, I want to read a statement from Thermo Fisher Scientific. Thermo is not endorsing, recommending, or promoting any use or application of Thermo products. Presented during the seminar, information and materials are provided by the third party, me, and without any warranty, including intellectual property or reported results. You can read the rest of it, but you can see that the presenting images have the rights to do so, and as part of this program, there's a fee from Thermo Fisher Scientific. So I'm very pleased to be here and really looking forward to interaction. I'm going to talk about the learning objectives, for which there are 4. And then we're going to have a few poll questions as part of our event, and hopefully leave at least a couple of minutes for questions at the end. So let me start with saying we've got 4 learning objectives. Number 1, describing the different types of tests that exist today. Number 2, the differences between those LDTs and IVDs. Number 3, listing the benefits of running an IVD and an LDT, and explaining how these 2 different types of tests are designed and regulated in the U.S. I hope that you're pleased with this. We're going to talk about all of that. Those are the core learning objectives. I'll also end this section and tell you that I will end the presentation talking about COVID tests and where they belong in the broad spectrum of IVDs, LDTs, and a new area called EUAs, which I'm sure all of you have heard about. So as I do that, let's talk about those 7 spellbinding questions. And the core here is first, all these question words: where, what, why, who, when, how, and can. So number 1, basics of diagnostic testing terminology. That's the foundation. I suspect for most of you it'll be a review, but we will start with there. We will then talk about an LDT and an IVD. Why do people use them? Who uses them? When can I develop these tests? How long does it take? What are the key parts to doing it? How do LDTs compare to IVDs? We'll be talking about throughout our presentation. And importantly, can a lab protect their intellectual property or IP, both for an LDT and an IVD? I hope that gives you a comprehensive sense of what we're going to talk about today. So first, let me start broadly with diagnostic test technology. And as you can see on the left, there are tests called in vivo tests. Most of the time that term is not actually used, but it means in the body or live. These are tests like physiology tests, an EKG, a blood pressure monitoring. All the imaging tests are typically called in vivo tests: X-ray, CT, MRI, PET. Most salient piece here is they're driven by physicians. Yes, there are automated systems, but it's a physician who signs off on those tests and typically is the person going forward to recommend that test. So that's one major category. If we look at the second category, it's called in vitro, and that technically means in glass. These are samples that are taken outside the body and analyzed. In vivo are samples of the body, and the mechanism, the biomarkers, always stay in the body. But in vitro, it's taking out a sample. So let's talk about the 2 differences, and I'm big into color here. So what you'll see is everything when I talk about IVDs will be in blue, and LDTs will be in maroon. So it gives you a sense of how we're going to go forward. So if I look at IVDs first, they're tests performed on bodily fluids, blood, urine, cells, tissues like a pap smear or a biopsy. they detect or quantify levels of a biomarker. Sometimes they just detect yes or no. Other times they say, let's quantify. Enzymes, proteins to diagnose cancer, bodily function disorders, cellular malfunctions. The key thing, and I'm going to go into detail on this in a moment, is they're driven by the products, from simple chemistry to ELISAs to flow cytometry. But what about LDTs? A lot of similarities. Number one, same sort of bodily fluids. Number two, same sort of biomarkers that we're looking for, enzymes, proteins, or otherwise. But the key here is the technology is driven by the process itself, not by the products or the tests. So let's break that down further. And again, IVDs on the left in blue. So I'm sure you're familiar with IVDs, but let me clarify some of the nuances here. They are developed for sale to diagnostic labs, health clinics, or consumers. If you're from a lab, you could develop an IVD, but most of the time it's manufacturers who develop the IVD, and then they sell it to labs. It's standardized instrument qualification procedures and training is required. The test itself and all pieces must be pre-validated, and a data analysis needs to be on the test itself along with a bioinformatics report. And critically, we're going to talk in more detail, keep peeling the onion here, the test must be clinically validated. So how, in this case, how does that change when we talk about LDTs? Well, here there are some real differences. Number one is LDTs are developed by individual labs and they are not transferred, licensed, or sold. This is a key issue where the FDA and all the regulatory authorities are very aggressive. You can't sort of say, I'm only selling it to one other lab. Sometimes even within the same institution, depending on your licenses, even within the same institution, it might be considered a different laboratory. It's critical to understand it's meant to be laboratory developed and stay in that laboratory. Here, the instruments need to be qualified as well, and training requirements must be established by the individual laboratory. Clearly, all the time you're going to have sophisticated people that are trained, but as part of the requirements for an LDT, you need to actually establish your training requirements as a piece of it. Here, by definition, they're developed in-house, and they don't need to have a standard assay to compare it to. Where with IVDs, when we get into regulation, you need to do that. And then lastly, they must be clinically verified. I use verified as opposed to validated because it's a slightly different process. Most of the time we talk about traditional LDTs, but given we're still in a worldwide pandemic, there are some exceptions here where they can be implemented quickly for emergency use, but they still need to be CLIA compliant, and that would be called an EUA. So that gives you the initial background. But let's now step up a bit and realize, how are tests being used? How can molecular tests for a wide variety of assays for different applications? And these can be, it's in blue, but these can be LDTs or IVDs. And if you look around this, I think it's a hexagon, what you could see here is 6 size. Genes and proteins, pathogens, gene expression levels, oncology markers, genetic variants, any of these, and it also includes pharmacogenomic markers, most often connected to a therapeutic and called companion diagnostics. So this is the industry writ large. Now let's go into the details as why develop an IVD. Well, the clear piece here is if you're a manufacturer, why do you develop these? Well, one is it is about broad distribution, increased revenue. It would surprise me if there aren't multiple labs in every small city, town around the world running IVDs. They're easy to establish as a gold standard, in part because there is a complete validation process. So if you look over here, they're easy to establish as a gold standard, in part because there is a complete clinical validation. That includes adverse event reporting as well as design controls and manufacturing controls. So it can be a very powerful test. And we're going to ask some poll questions later, but many of you very well are using those today. But how does that contrast with an LDT? Well, here what we see is an LDT is very different. First, it's because there's no IVD test available. Now, sometimes there can be something similar available, but not exactly the same. And that's very much related to the LDT would be an improvement over the existing assay. And no IVD aligns with the procedure that you want to use. That is why it starts. But how are they used? Well, they must be developed, and they're likely to be developed more rapidly than an IVD. One of the key pieces, and a great example here, is in the AIDS environment. When we were first understanding AIDS, the initial tests were LDTs because we didn't always know exactly what to test. So we were able to put together, as an industry, tests and then continue adding analytes. That is true of breast cancer. That is true of a number of different tests, that it can be more rapidly developed than an IVD. In addition, it can help a lab be competitive. Why is that? It's because it's an improvement over existing assay, and it can be added to over time in a relatively quick fashion. So this is a key piece of why developing an IVD can be essential. So what are the LDT. But now let's move back to IVDs. What are the advantages of them? We talked about a few of these, but I'm going to highlight them so you see them in a little bit more detail. I believe there are 5 of them. Number 1, a quality system. The manufacturer has put together a rigorous quality system approved by the FDA, everything from design controls to manufacturing to handling complaints. Simplified inventory control for the laboratory itself. You only need to order what you need, as opposed to IVDs where you've got to order all the parts. It's sort of, as you're probably familiar, it's sort of like, you know, buying a whole car as opposed to buying a kit. Well, bad use of it. So buying the pieces to create your own car. Technical support. This is critical. The experienced laboratorians know there's always going to be problems. So with an IVD, you can go to the manufacturer to troubleshoot. We then have clinical validity. The clinical validation of the test is key and required by the FDA to ensure that it identifies, measures, and predicts the clinical condition prior to marketing. And then lastly, as a manufacturer, you get broad distribution and that it gets out to all the labs. So that sounds good, but if you're a lab and you're saying, okay, I run IVDs, but I want to do something unique because I see the need. So now let's move to the advantages of LDTs. Again, I think that there are 5 of them here. And what you could see is number 1, and this is really from the laboratory perspective, you own the content. You have control over that content. You can select specific and relevant targets. You can put into that test what you believe is necessary for the clinical market. Secondly, as I've talked about, they can be developed quickly, yes, but I think it's almost more important to say they can be modified relatively quickly. We'll talk about what it takes to modify them, but that is possible. So you can respond to market needs like a pandemic or in rare diseases. Third, this is interesting, lowered cost per test. I believe that is the case most of the time. I want to emphasize that some highly complex LDTs may be more expensive than a very simple IVD. So you can't say, you can't generalize this about every LDT as a lower cost per test. But for the most part, you're buying the individual pieces of it, the equipment, the reagents, and you're putting it together. The cost comes in the labor that happens in your lab, the R&D, etc., not typically in the pieces you're buying from outside. Next, consolidating into a single test. And this is connected to control over content right there. What that means is, let's say, you know, this happened in the prenatal area. There were a number of different IVDs, but with a precious small sample, because you don't want to get too many samples, whether that would be from amniotic fluid or blood, you create an LDT from existing IVDs. So it gives you the opportunity to put multiple analytes together and enable potentially a faster diagnosis. And lastly, it's all about the lab. If there's one key piece to LDTs and lab qualifications, what you are doing is qualifying your lab, not individual tests. So this is a fundamental and foundational piece, especially if you expect to be doing multiple LDTs in the future. So what is the process? What I'd like to do, and I'm going to emphasize this one way or another, but before we get to this, you should see on the right-hand side of your scale, on your screen, a question. And I'm going to read the question, and I'm going to ask you to answer the question, go into the little circles next to the A, B, C, D. What kind of diagnostic tests are you running in your lab today? A, in vitro diagnostics. B, laboratory developed tests, as we've just talked about, both or neither. So I'd ask you to go in and again check mark in the circles and hit submit. I'm going to give you a moment to read the question and go do that, and then I'm going to continue on to my discussion. I am indeed going to share the results with all of you. So as you see on the screen right now, I believe there are 5, really, we'll call it 6, different parts of an LDT process. And the first critical part is all about planning. It's all about that lab setup. Yes, you need CLIA certification. Most high-quality labs also need CAP certification, but that takes a little bit longer time to do. JCO for academic labs, and important to recognize that while CLIA is federal, it's the state-specific regulations are also apply. I'm sure if you've been in the laboratory industry for any period of time, you know that New York has very stringent requirements, more stringent than the federal CLIA certification. For those who haven't been in the industry, it's why sometimes labs will say they're in 47, 48, 49 states, and most of the time New York's the last one. So then we move on to planning. And in moving on to planning, you can see that there are a couple of key drivers. I'm going to go to the poll in a moment and talk about that, but I want to finish the planning piece because I think it's very easy to say, you know what, I'm just going to skip the planning aspect of it, but it's critical. And we can see from our poll that a lot of people are pretty familiar. 21% of you run LDTs, same 21% of you run IVDs, 29% are both, so you're familiar with it, and 14%, I'm assuming, are in different parts of the industry and are not running either test, either now or on a different part of the value curve. So let's go back to this. Where do we go first? It's up here in planning. So when we talk about planning, you need to understand all of that, including reimbursement. I'm not going to talk about reimbursement today, but ensure that you know what it's going to cost for the technical requirements, what it's going to cost for the operational improvements, and what you're going to get back in reimbursement. You then move on to the core, but I think the so what test. What is the test menu? What are the targets? How do you customize it? How are you going to interpret it? And I'm going to emphasize here reporting. I can't say enough about this. So many people creating tests, they're so excited about it, and they didn't think through what the actual report is. And it says, well, we can do that later. Well, I just have to tell you, you know, Genomic Health is incredibly successful, one of the most successful companies in the industry. they launched a great test, and the report was hard to understand. And as a result, they had to take it off the market and relaunch. I mean, this is now 20 plus years ago, but don't forget about reporting, and don't forget that the #1 area today of malpractice cases, or #1 or #2, is the doctors didn't understand the report that they got from the laboratory. That is critical. We then move on to AV, or analytic validation. Here, you're probably familiar with these, but CLSI is the key government or a key governing body, and you need to do all of these: sensitivity, specificity, reproducibility, etc. And interference tests to know what could possibly interfere with an accurate sample. You then move on to clinical verification. I get a lot of questions about this because in IVDs it's pre-subscribed. In LDTs, it's important to work with CLSI and understand how many samples are necessary depending on the technology you use in your test. So that's a key piece of that. And lastly, go to green. It's all about test launch. And while there's lots of things about test launch that you need to be thinking about, what is your distribution strategy? I'm going to highlight 3. One is getting your tests to providers that they understand how to use it. #2, understanding and describing the test utility. And #3, if your IP strategy allows, publish, 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 publish. That is the best way to, even in a pandemic, and maybe more so in a pandemic, to get the information out about your test. So that gives you the overview of the process, but what's happening on the regulation? And I've kind of hinted at a few of these, but I want to go into a little bit more detail about this. Again, in blue on the left, what we have here are diagnostics are included in the FDA definition: reagents, instruments, systems intended for the use to diagnose disease or other conditions. And you can read the rest of it, but it's about curing, mitigating, treating, or preventing disease. These IVDs, as we've talked about, are classified as a device. We can debate whether that's right or wrong, but it's true today. They might require a PMA, or a sponsor can use a 510K, typically, if there's a predicate device that exists. Basically, another device, i.e. a diagnostic, that is measuring what your test is measuring. The sponsor, i.e. you, the laboratory, you can use non-approved, non-cleared FDA IVDs, but those are only for research use only or investigative use only, RUO or IUO. But once again, these are one of these key highlights to remember. You have to use proper labeling, and you cannot use it for clinical diagnostic procedures. Another area where the FDA is very aggressive. And I've seen a lot of labs get into trouble that they've been using an RUO for 10 years. It's the standard in the industry. Everybody talks about it. Everybody knows the tests. And then it kind of ends up in clinical diagnostic procedures. You need to make it sure it's an LDT or IVD, and it is not classified as an RUO anymore when you're using it for clinical. And lastly, everybody's favorite 3 letters, well, maybe FDA and EUA. We know that the FDA has authority to do that. So let's contrast all of that over here with an LDT. So #1, the FDA says it could regulate LDTs, but they're exercising enforcement discretion. We are not going to get into the politics behind this, but it's important for you to know that it might change. The FDA has said if it does change, there'll be adequate time for transition, but right now they do not regulate LDTs. They are regulated under the 1988 CLIA amendments, the Clinical Laboratory Improvement Act, and all laboratories that test patient samples must obtain the Certificate of Compliance or accreditation to bill CMS, the Center for Medicare and Medicaid, for their services. Technically, You could have a lab without CLIA. Some payers might reimburse you. I'm going to tell you, don't do it. It's not the right way to go, and payers really shouldn't reimburse you. But what I said before, CLIA focuses on the lab and the personnel, not the test. So you could have 10 LDTs. You need to validate and verify the processes, but your lab is what goes through CLIA. And when it says personnel, it means personnel. It means what your people are wearing, how they're being trained, what experience they have, whether they're wearing open-toed shoes, etc. LDTs and IVDs are classified by different levels of complexity. We'll get to that. And in terms of EUAs, at the beginning of the pandemic, the FDA was saying that LDTs for COVID needed to go through the FDA EUA process. However, HHS stepped in August and said that isn't true. So it changed. So let's talk about those test complexities. There are 3 different areas: waived, moderately complex, and highly complex. And often just called, you see W, M, and H, but just these are often just called waived. These are called moderate and just high. Pretty obvious. What's the description? Simple to perform, low risk of error, interpretation error. And any of these are now sold over the counter. These are very simple tests. You'll often find them in doctor's offices where they'll say the doctor has a POC, a point of care lab. It's done often at the bedside. These are usually quick tests that get answers quickly. Secondly, moderately complex tests. These are usually performed with automated equipment, and you can see the list of it here. I think and these are probably the most common tests that you see: a CBC, a complete blood count. Interesting, you can have a urine test here with just a dipstick looking for sugars, and then a urine drug screen here. So the sample itself is not relevant. You can have the same sample in each of the categories. Lastly, a high complex test requires expertise more than just the automation and may require more data analysis, particularly something like NGS. Next-gen sequencing requires, data analysis more than just the laboratory itself. You can see a list of these here, and clearly RT-PCR. For some of the tests in the pandemic, they've moved to moderately complex, but typically they're over here in terms of highly complex tests. So this gives you a sense of it. I think this classification is here to stay, even if there are other changes, so important to recognize the difference here. And this category of OTC becomes more and more important. So as we do this, we're going to move to our next section, which is intellectual property. And there's another poll question. You guys did a great job before. Thank you. I'm now going to call your attention to the right side of your slide. A simple question. Have you attempted, or are you attempting, to obtain IP for your LDTs? So if you're doing LDTs, we would appreciate knowing how important IP protection is, and is this something that you look for or don't look for? So again, you guys got this. Fill in the circle and press submit. So I'm going to share 2 slides on intellectual property, and I like to think of them, I'm a football fan, as offense and defense. It's not quite as simple as that, but I'm going to start with offense, proactively obtaining IP protection. And this is challenging because of 2 things. One is, if you're using traditional platforms, you're buying platforms, they're really, really powerful, very, very important, but hard to get IP if it's somebody else's equipment. You probably know that. Secondly, the use of known or previously published biomarkers. That is a key. But that does not mean, and I'm going to talk about some legal cases around this, it does not mean that you shouldn't look or at least consider it. So first, let me start with, you might get IP protection on novel platform technologies. You might get IP protection on improvements to conventional platform technologies, although I'd say work with the manufacturer and ask about that, so to make sure you understand where they are. But then the area that most people talk about are novel biomarkers. And in 2012 was one of the most important cases in terms of LDTs that the Supreme Court has ever dealt with. It's called Mayo versus Prometheus. Mayo is in Mayo Labs, Prometheus as in a diagnostic lab provider. And Prometheus had a test for drug metabolism. They had the IP for this test, and Mayo challenged them. And Mayo was running essentially the same test, and they said, Prometheus, you can't have IP protection of that, and we're just going to run the test. Well, the Supreme Court agreed with Mayo, and what they said was that Prometheus was not able to patent that, so they invalidated the patent that Prometheus had. Many of you are also familiar with a more recent case, which is Myriad versus AMP. And in the Myriad case, it was all about the fact that you can't patent naturally occurring events, particularly DNA, and they were looking to patent the mutations that they understood the significance to in the BRCA gene. I don't highlight that quite as much because that has to do with everything from therapeutics to diagnostics, but those are two key cases. So if you choose not to go forward with patent protection, or you try and the answer is probably not going to happen, think about trade secret protection. Very powerful, and doing it right and ensuring that your team understands that is key. So that's offense. Now let's go on the defense, but I'm going to call it sort of proactive defense. What should you be thinking about when you're developing an LDT? Well, regardless of the issues with Mayo, patent infringement is still a concern. So the last thing you want to do is infringe on somebody else's patent, certainly not have them infringe on yours. But so if you're starting this, part of your planning should be considering a freedom to operate opinion. Now this is not cheap to get. So some labs say, I'm not going to get it now, I'm going to wait till I get funding, I'm going to wait to know whether the test works, which is great. But it's important to at least consider this, particularly if you're taking this test out of an academic lab and creating an individual company focused on this. I think you have the overview of the IP, and we can see in our poll that 68% of you are not focused on IP for LDTs, 19% of you are. I'm not surprised by those numbers, but whenever you do, hopefully this gives you a sense of what's out there and some of the key pieces of information you should take into account. So with that, I'm going to close this section and I'm going to talk about LDTs in a different context, which is LDTs in the midst of COVID-19. And I don't, there's probably hardly anything that has not been said about LDTs. But I'm still going to say a few more things and hopefully put the data together in a way that might be interesting to you, and we'll have one more poll question. So you know that the CDC developed an LDT for SARS-CoV-2. It did not go terribly well. We can argue as to why or how or otherwise, but the bottom line is it changed. After that, University of Washington and Broad developed their own LDTs by the end of January 2020, and the genome for SARS-CoV-2 was developed, I believe it was January 19th, and people developed LDTs in a ridiculously fast time. I didn't talk about timing before, but when we talk about LDTs broadly, it's probably a minimum of 6 months, more likely 9 to 12 months. The idea that these happened in 10 days is crazy, but we know lots of things happened out of necessity in COVID-19 that we would not have expected. So US Labs also developed an antibody-based assay, but I'm going to focus most of my discussion on the active virus testing, PCR, and antigen. So you guys know this, but I'm going to put it in for completeness. What can we look for when we look for COVID-19 tests? Number one, the virus itself. Number two, the infected or the immune reaction. You know the virus, it's the mRNA genome or the viral proteins. These are the PCR tests, and these are the antigen tests. And it's all about the external structures for the antigen tests. When we're looking at the infected, not whether somebody is infectious with the viral elements, this does get more complicated. And I believe because of this complication is why antibody tests have not done well in the U.S. There are really three things that we're looking for, is B-cell, circulating antibodies, circulating activated T cells, or systemic biometrics. And this is a wide range, but I'm going to focus on the two of these. When we're looking for this immune reaction to a vaccine, most people are talking about circulating antibodies, neutralizing antibodies, and those are based on the B cells. I think the reason this is not done well broadly is because you could have activated T cells to a vaccine or to a previous infection and still be protected. But everybody interprets this very differently, but I thought it was important to understand the different aspects of COVID tests from a baseline level. So let's talk about, not animated, but let's talk about the history and progression of COVID diagnostics. We start at the top here, and I'm going to look at it in 4 different criteria. One is it started that it was only symptomatic diagnosis. You literally needed to go into the operating room to have a bronchial lavage. We then moved to active disease diagnostics with PCR or antigens, then screening, and I hope soon we're going to move into the surveillance part, air monitoring, breath tests, sniffing dogs, scratch and sniff tests, and most importantly, wastewater testing. First and the last, well, everything is real, but the first and the last I have the most confidence in terms of surveillance. This is critical. And where do LDTs and IVDs fit in? Right here, right in the middle, active disease and screening. So now let's look to where these tests are being done. They started in hospitals, moved to the central lab, now moving to point of care and individual labs, and then to home and self-tests. But are these tests difficult? You know that from invasive to semi-invasive to minimally invasive to passive. And what is the price? Thousands to hundreds to tens to less than $5. So now that you have an overview of COVID tests, I'd love you to go to the poll, our last question. If you are running COVID tests, which ones are you running? Select all that apply. So are you running PCR, antigen, whether that's handheld or instrument, serology, or if you're not running any COVID tests? I thought it was important to give the history. I'm sure many of you know this and are familiar with it, but I find it very unfortunate that many still believe that testing requires nasopharyngeal swabs and that that's the only way to do testing, because we now know anterior nasus and mid-terminate swabs are equally effective, as is saliva. So many people are stuck where we were over here. So what are the tests out there? I'm going to continue and then I'll share the poll results in a few minutes. What are the tests that are out there? This gives you a sense. This is from 3 months ago, and in the next week there'll be an update here. But it just gives you a sense. The FDA has approved just over 400 tests. The IVDs in Europe, 750 tests. LDTs before that stopped for about 300, and then you could see development. It's important to recognize that the FDA does have about 180 tests that were revoked, rejected, withdrawn, warning letter, or fraud. This is a big deal. Just because a test got an EUA doesn't mean it stays with that. And now I know you know that because I'm looking here. 58% of you are not running tests today. The largest are running PCR, not surprised, and about the same number are running antigen and serology, and some running both. So let's take a deeper look at the FDA EUAs. This is through June, but the numbers and the trends have stayed very much the same. Molecular, the largest, again here in blue, 88% of those are PCR, only 1% CRISPR, and 3% sequencing. Antigen, overwhelmingly lateral flow. Antibodies, split about 2/3, 1/3 between ELISA and lateral flow. And then the FDA has issued EUAs for collection kits, and there are only 6 tests for patient management. But what about the future? If we look at testing development worldwide, not just the US, you remember from this slide, going back, 88% PCR, 3% sequencing, and CRISPR. Let's go back here. Only 40% of development is in PCR, a chunk in isothermal, and now we have a full 30% in CRISPR and sequencing. I think this will be the key area in the future. The rest, really no surprises, except over here where we have breath tests, mass spec, and Raman spectroscopy. We don't have any of these tests approved, despite the fact there was a lot of buzz about them, but they haven't gone through yet. Why is this all important, and why are we still testing Delta, Delta, Delta? Delta accounts for virtually 100% of all tests in the US. Give you a sense of how fast. It took 1 month to be 51%. Alpha, originally called the UK, variant took 3 months to go into dominance. It moves fast, and sadly, it's a pretty perfect virus. What does that mean for testing? Well, I have 5 questions and what I call my so what questions. First of all, this is University of Utah electron microscope, a great picture of a SARS-CoV-2 cell, and these green things, as you know, are the spike proteins. So what about the variants? What are my 5 questions? Number 1, does it make the test less accurate? No. Tests work. Antigen tests and certainly PCR tests. Number 2, does it increase cases and deaths? Sadly, yes. Does it make some treatments ineffective? It does make some treatments ineffective, particularly around monoclonal antibodies. If somebody recovered from alpha, you can't use their antibodies to treat somebody with delta. Does it reduce vaccine effectiveness? It does. And that is one of the saddest issues today. However, I want to emphasize the fact that while it reduces vaccine effectiveness in terms of breakthrough cases, the vaccines remain 95, 97, 99% effective against serious disease and death. And then lastly, does it raise the hurdle for herd immunity? It does. Whether we'll ever get there today, I don't know. What I do know is that our response has to be tests as well as vaccination. So how, this is very relevant just this week. When you talk about, well, we sadly saw that the U.S. has now had more COVID-19 deaths than the 1918. I actually get emotional talking about it, the 1918 flu. If you had asked me a year ago, I would never imagine that with all the technology we have today, that it's different. Now, clearly, there are more people in the U.S. today. So while COVID killed 1 in 500 today, it was 1 in 150 in 1918. But still important to recognize the differences. And for many, many, many of these, there are IVD and there are also LDT tests. Most recently, Ebola, where there were tests. And what you can see here is comparing case fatality rate to the R0, or transmissibility. So you have a lot of diseases here. And unfortunately, you can see the difference where this was the original Wuhan strain, where the average person only infected 2, and here we are with Delta with 6. And that is a concern. But what we don't want to do is go up this ladder. Ebola was a pretty effective virus, but since it killed so many people that it infected, it actually couldn't live long. The only two that are above it are rabies and HIV, but we know how to treat those. So this is very, very critical to understand where we are. There are diseases, and I hope we never go here with the mutation from COVID that is more infectious, but indeed measles, mumps, whooping cough, malaria, and if you've been on a cruise, rotavirus, are highly contagious, but the death rates are low. So with that, I want to get to my conclusion, and I think we have a couple of minutes to take some questions. LDTs and IVDs are central to clinical patient care as well as research. Both types of tests help with personalized medicine. LDTs are critical for diagnostic innovation. They leverage a different regulatory system that allows the labs to adapt quickly. And LDTs are tied to laboratory processes where IVDs are tied to laboratory tests. So thank you for your interest and attendance, and I'm happy to take some questions in the time that we have.